My name is Josh Mann. I am with QIAGEN. I've been with QIAGEN for about 3 years now. So I'm going to discuss the QIAcube. I'm going to let you know what it's all about, discuss some of the key features, some of our internal validation data, as well as some application data, and give you a summary on the system, as well as some maintenance recommendations for the instrument. So what is the QIAcube all about? DNA structure was determined and published in 1953. After that, there was purification and isolation techniques that were developed. In 1993, QIAGEN came out with the mini-spin column, the membrane that we've been processing samples on, a similar column. In 2007, QIAGEN developed the QIAcube, which was sort of the natural progression for processing these mini-spin columns. So it's really the next logical step when processing, your samples in the lab, RNA, DNA, protein. It's become sort of a routine procedure, something that everybody's using in the lab. So, as it says here, there's city maps were used, abacus, washing dishes, and, now we use instruments to perform these tasks. And the QIAcube sort of takes these same type of manual tasks and does it within an instrument and standardizes your results. So do you ever think about how much time you spend processing your samples in the lab? Takes a while, huh? A typical lab on average spends an hour and a half per day. I know many of you know that could be a lot more. 200 days per year, basically 2 months of continuous prepping in the lab. Yeah, when it puts it in that perspective, it's a little daunting. So this is routine, everyday work that, if possible, if you could have an instrument that could do that for you, it could save a lot of time and also, standardize what you're doing in the lab with these processes. And also, what is your error or failure rate? And that's something I don't think that's really quantified and isn't necessarily, you aren't able to really quantify what your failure rate is because you don't know what necessarily goes wrong with your samples when there is a mistake that goes on. And can you use your time more efficiently? And I'm sure all of you would say yes. So with the progression from manual to automation, basically you're eliminating manual sample purification and increasing your efficiency, using your time for other things, whether it's writing articles, designing new experiments, and doing your data analysis. The shift from manual to automation processing of your samples basically gives you the safety and convenience of using a proven chemistry, as well as flexibility within the platform that you're using, and making you more efficient and standardizing your results through automation. So I'm going to go through some of this a little quickly because we went through a lot of this in the lab yesterday, but if there's anything you'd like elaborated on, definitely feel free to stop me and we can go into things a little bit more in depth. So the QIAcube is the automation platform that we're going to discuss today, and it's able to do DNA, RNA, and protein purification, as well as the cleanup steps. DNA cleanup, RNA cleanup. It's a platform that offers an easy download and transfer of new protocols. There's a load check before each run, and it's a small benchtop system, 2 feet cubed, and it has a centrifuge, a heated shaker that's built in within the instrument. As we saw with the QIAcube, there's the load check that, so if anything's out of place, if you don't have enough buffers, if you don't have your tubes are out of place where the centrifuge won't be centrifuge rotor won't be balanced, it will give you an error and let you know where that error is and prompt you to fix that. So it's sort of a fail-safe mechanism so to help prevent errors. This is a screenshot of the inside of the QIAcube. As you can see, there's a centrifuge rotor. There's rotor adapters where you do your spins, your washes, and the rotor adapters catch your washes. There's in the number 3 position is your sample rack. The number 4 position is the rack for your buffers. Number 9 is where the gripper head mechanism is located, so that will move your spin column into your elution tube, as well as that's where the pipetting head is located. What the QIAcube does is basically takes the same processes you use for processing the same mini spin columns that you would do manually and does them within the instrument. It uses the same kits, it uses the same buffers, it just takes the same protocol and does it within the instrument without the human potential for variation of your data and your yield. So 3 simple steps that the QIAcube does is it loads your samples, you load your buffers, and you load the rotor, and you're ready to go. As you saw yesterday, you press go, and 30 minutes later, we were able to pull out our cleanup samples. Once everything's in place and you have it initially set up and you get used to running it, as we saw with the second group, it was, all the buffers were in place, so it was really maybe 5 or 10 minutes, I think, setting it up, and then we pulled our samples out. So it has the touch screen. So when you're setting it up to go, you basically pick the protocol that you're running. It's pretty user-friendly, so whether you're doing RNA, DNA, protein, or cleanup, you press the screen for the specific protocol you'd like to run and go into that protocol within the cleanup DNA or RNA list and press start. So as I mentioned, there's a load check before you start. Verifies the number of samples. The instrument is fairly intuitive, so it will let you know if you have enough buffers within the instrument to process the amount of samples that are in there. It'll let you know to add more buffers if you don't have enough in there. Also checks the amount of tips that are in the instrument, and if you don't have enough tips in there, it'll also prompt you to add more tips. Believe now there's over 100 protocols that are available on the QIAcube. Anywhere, DNA, RNA, protein, you can process a variety of samples on the QIAcube. One aspect of the QIAcube that I think complements it fairly well is the web portal. So we didn't really go into that yesterday, but on our website you can go into the QIAcube web portal. You can pick out specific protocols if you want to, do a different kind of sample. So we have a QIAmp DNA Investigator kit that's available for the QIAcube. And you can download that protocol, put it on a USB stick, and then there's a plug-in in front of the QIAcube and upload the protocol onto the QIAcube. So you can add and remove programs, as needed. Also, if, as we discussed, if you wanted to add 2 or 3 washes to your protocol, we could program that protocol, make it available on the website, and then you could basically just go to the website, download the protocol, and then upload it onto the QIAcube, and you have that protocol available. So it's a nice tool to use with the QIAcube, and all of our protocol sheets are available on there as well. So that cheat sheet that we used with the QIAcube, where it lets you know where to put everything on the deck and how to arrange everything, you can download those protocol sheets as well, in addition to the protocols from the web portal. So we were using the QIAcube yesterday for MinElute cleanup. It's able to process anywhere from 2 to 12 samples on our mini spin columns. As we saw, it can greatly enhance forensic casework analysis, increase your profile, visibility of your profile. Let's see here. The MinElute PCR purification kit substantially improves performance in casework DNA profiling, as we saw earlier with the data that we went over. Higher signal-to-noise ratios, sensitivity, and allele call rates. Superior results compared to filtration or enzyme-mediated digests are made possible through these mini spin columns. Recent data suggests that the standard cycle number PCR followed by post-PCR purification delivers results equivalent to increased cycle numbered PCR. As Matt was going over yesterday, this Foster et al. Paper shows that the combination of the 28 cycle PCR with post-PCR purification using the MinElute PCR purification kit provided equivalent results in STR analysis, reduced sample consumption, reduced stutter peak ratios, and reduced the frequency of non-donor allele peaks. Now this is a comparison of the QIAcube as it compares to the Easy One and QIAsymphony. So some of you I know have the Easy Ones, and the difference between the QIAcube chemistry and the Easy One chemistry is basically that same silica membrane that we're processing our samples through on the column. It's just a silica membrane wrapped around a magnetic bead. So this just shows, I mean, the question is, well, how does this compare to, the silica membrane versus the magnetic bead? So you can see there's linearity between the two chemistries and the various platforms. So QIAcube is able to process 1 to 12 samples. The Easy One Advanced is a bead-based technology. You can process either 1 to 6 samples and now 1 to 14 samples as well on the new XL unit. And then the QIAsymphony is also another bead-based chemistry. Or it runs a bead-based chemistry, and you can process anywhere from 1 to 96 samples. As far as validation data, there's other kits obviously available that you can run on the QIAcube in addition to MinElute, and we have an investigator kit that you can run on the QIAcube, similar, as we saw from the data on the previous slide, to the kits that you can run on the Easy One. So there's, for a forensics lab, there's the investigator kit, there's post-PCR cleanup available on the QIAcube. Now, as far as developmental validation, when testing the DNA investigator kit, there was a variety of samples that were put on the QIAcube and processed, sort of forensic type samples. And so next slide we'll see, let's see here, compared also manual and automation extractions. We used the FTA paper, and what you can see is it's fairly similar results comparing the manual and automated extractions. And one thing we saw yesterday and then we continue to see is obviously when you're running on an automated platform, when the pipetting variable is standardized, as well as the amount of time that your sample might be sitting on a silica membrane and everything, you're going to get more standardized results. So there's going to be less variation between runs and also between samples because your pipetting is obviously being a lot more controlled. So there's linearity between your processing of samples. So in a dilution series of blood, saliva, and cultured cells, linear input to yield correlation, So we can see with the 3 different sample types with varying input material, it's linearity with your yield. And as far as sensitivity, signal to noise, everything looks good, going down to 0.1 microliters. Full profiles for all the dilutions, low noise levels. Also with our internal validation, we did a lot of cross-contamination testing. The way the arm moves within the instrument, we wanted to make sure that there isn't any crossover of samples. So, you know, you don't want to, if there was, for whatever reason, if there was any drippage or anything, you don't want the potential for contamination. So we did a lot of checkerboard type format of testing where we, you know, do every other sample and then, you know, do blanks and every other, similar to what Rob was discussing on what he did on his testing of the MinElute kit. And didn't see any contamination. It changes your tips between samples, and there's no crossover over your buffers or your sample tubes. So for the cross-contamination testing, 100 microliters of human whole blood was used. 4 consecutive runs with alternating pattern of positive and negative samples, checkerboard format, 48 samples total, downstream analysis using the real-time PCR, and uniform yields from the 100 microliters of blood samples, no human DNA detectable in mock sample eluates, distribution of yields across run, you can see there on the left side, looks good. So where does the QIAcube fit into the forensics workflow? It has the capability of being used for sample purification. So it's a little bit of a different application than or utilization than the Easy One Advanced or the QIAsymphony. And then as we've been going into yesterday and today, it can also be used for post-PCR purification. So this was a little bit more testing that was done with just cell culture, just a comparison of the QIAcube's yield versus manual. And it's just an overnight, you know, cells that were grown up. And there's linearity between manual and QIAcube sample processing. And this testing was done with our RNA prep kits as well. And this is using the QIAmp-DNA blood mini protocol. Purification of highly pure DNA is seen. There's 3 donors, and you can see with the different anticoagulates added, the yield looks good. I'm going to go into the maintenance of the QIAcube now. Really, the maintenance of the QIAcube is fairly minimal. After each run, you want to empty the waste drawer, remove any disposables that you don't need anymore, replace the lids, store the bottles according to the instructions on the kit, and you can either run another protocol or turn off the QIAcube. As far as monthly maintenance, we just recommend wiping it down with a detergent, and that's really it. I mean, it really doesn't require a lot of maintenance. Also cleaning the optical sensor, so that's the sort of the laser that we kind of saw going around the deck to make sure everything's in place. We would recommend wiping that down once a month, and just wiping the gripper head and the different and the pipetting head, the different pieces of machinery that are within that arm that moves around. So, and that's about it for the maintenance. You want to clean the shaker rack, and clean everything out with distilled water and an appropriate cleaning agent. And obviously clean the waste drawer. So as you can see, the QIAcube increases your efficiency by elimination of routine tasks, uses a proven chemistry, and offers flexibility of processing 2 to 12 samples, and gives you the reproducibility by automation of the complete process that you would be doing at the bench. Any questions? Yeah.
How is this robot being checked at all, like pipetting wise, like how accurate your pipette is?
Yeah. So you can have a maintenance done every, a yearly maintenance where they will check the pipetting, but before it's shipped to you, that's part of our quality control that's done before it's shipped out, to a customer.
So that could be part of like the yearly PM, or would it be something additional that you would ask for, or do you automatically do it?
It should be part of the yearly PM.
Josh, you talked about the validation studies that you did at QIAGEN. Are those on your website, or are they results of those relevant assessments?
Those probably aren't, but we do have some, probably a little more useful for this group. We have some validation testing that was done for a couple different forensics departments around the U.S. that they've done when they've gotten the QIAcube up and running in their labs. So if anybody was interested, if they were in the process of validating a QIAcube and wanted to let me know, I could get what those labs done or did into your hands, and you could maybe duplicate or write something similar up for your validation.
Those drives that you have some of the papers, like the Smith and Valentine paper, the ones that have been left.
Yeah, and the Ballantyne lab is actually using just the standard MinElute protocol now too. I know in his original write-up he was doing, I think, the 4 washes, and then they ended up getting a QIAcube, and so I know they're doing the standard protocol that comes on the QIAcube that we ran yesterday.